Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Cytosol. 51 languages. The cytosol is a crowded solution of many different types of molecules that occupy up to 30% of the cytoplasmic volume. 1. Cell biology. Animal cell diagram. Components of a typical animal cell. 1 nucleolus. 2 nucleus. 3 ribosome, dots as part of 5. 4 vesicle. The cytosol, also known as cytoplasmic matrix or ground plasm, 2, is one of the liquids found inside cells, intracellular fluid, ICF. 3. It is separated into compartments by membranes. For example, the mitochondrial matrix separates the mitochondrion into many compartments. In the eukaryotic cell, the cytosol is surrounded by the cell membrane and is part of the cytoplasm, which also comprises the mitochondria, plastids, and other organelles, but not their internal fluids and structures. The cell nucleus is separate. The cytosol is thus a liquid matrix around the organelles. In prokaryotes, most of the chemical reactions of metabolism take place in the cytosol, while a few take place in membranes or in the periplasmic space. In eukaryotes, while many metabolic pathways still occur in the cytosol, others take place within organelles. The cytosol is a complex mixture of substances dissolved in water. Although water forms the large majority of the cytosol, its structure and properties within cells is not well understood. The concentrations of ions such as sodium and potassium in the cytosol are different to those in the extracellular fluid. These differences in ion levels are important in processes such as osmoregulation, cell signaling, and the generation of action potentials in excitable cells such. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 5 rough endoplasmic reticulum. 6 Golgi apparatus, or Golgi body. 7 cytoskeleton. 8 smooth endoplasmic reticulum. 9 mitochondrion. 10 vacuole. 11 cytosol, fluid that contains organelles, with which comprises cytoplasm. 12 lysosome. 13 centrosome. 14 cell membrane. As endocrine, nerve and muscle cells, the cytosol also contains large amounts of macromolecules, which can alter how molecules behave, through macromolecular crowding. Although it was once thought to be a simple solution of molecules, the cytosol has multiple levels of organization. These include concentration gradients of small molecules such as calcium, large complexes of enzymes that act together and take part in metabolic pathways, and protein complexes such as proteasomes and carboxysms that enclose and separate parts of the cytosol. Definition, edit. The term cytosol was first introduced in 1965 by H. Alardi, and initially referred to the liquid that was produced by breaking cells apart and pelleting all the insoluble components by ultracentrifugation. 4. 5. Such a soluble cell extract is not identical to the soluble part of the cell. cytoplasm and is usually called a cytoplasmic fraction. 6. The term cytosol is now used to refer to the liquid phase of the cytoplasm in an intact cell. 6. This excludes any part of the cytoplasm that is contained within organelles. 7. Due to the possibility of confusion between the use of the word cytosol to refer to both extracts of cells and the soluble part of the cytoplasm in intact cells, the phrase aqueous cytoplasm has been used to describe the liquid contents of the cytoplasm of living cells. 5. Prior to this, other terms, including hyaloplasm, 8, were used for the cell fluid, not always synonymously, as its nature was not well understood. See protoplasm. 6. Properties and composition, edit. The proportion of cell volume that is cytosol varies, for example. While this compartment forms the bulk of cell structure in bacteria, 9, in plant cells the main compartment is the large central vacuole. 10. The cytosol consists mostly of water, dissolved ions, small molecules, and large water-soluble molecules, such as proteins. The majority of these non-protein molecules have a molecular mass of less than 300 the 11. This mixture of small molecules is extraordinarily complex, as the Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Intracellular fluid content in humans. Variety of molecules that are involved in metabolism, the metabolites, is immense. For example, up to 200,000 different small molecules might be made in plants, although not all these will be present in the same species, or in a single cell. 12. Estimates of the number of metabolites in single cells such as E. coli and Baker's yeast predict that under 1,000 are made. 13. 14. Water, edit. Most of the cytosol is water, which makes up about 70% of the total volume of a typical cell. 15. The pH of the intracellular fluid is 7.4. 16. While human cytosolic pH ranges between 7.0 and 7.4, and is usually higher if a cell is growing. 17. The viscosity of cytoplasm is roughly the same as pure water. Although diffusion of small molecules through this liquid is about fourfold slower than in pure water, due mostly to collisions with the large numbers of macromolecules in the cytosol. 18. Studies in the brine shrimp have examined how water affects cell functions. These saw that a 20% reduction in the amount of water in a cell inhibits metabolism, with metabolism decreasing progressively as the cell dries out and all metabolic activity halting when the water level reaches 70% below normal. 5. Although water is vital for life, the structure of this water in the cytosol is not well understood, mostly because methods such as nuclear magnetic resonance spectroscopy only give information on the average structure of water, and cannot measure local variations at the microscopic scale. Even the structure of pure water is poorly understood, due to the ability of water to form structures such as water clusters through hydrogen bonds. 19. The classic view of water in cells is that about 5% of this water is strongly bound in bisolutes or macromolecules as water of solvation, while the majority has the same structure as pure water. 5. This water of solvation is not active in osmosis and may have different solvent properties, so that some dissolved molecules are excluded, while others become concentrated. 21. However, others argue that the effects of the high concentrations of macromolecules in cells extend throughout the cytosol and that water in cells behaves very differently from the water in dilute solutions. 22. These ideas include the proposal that cells contain zones of low and high density water, which could have widespread effects on the structures and functions of the other parts of the cell. 23. However, the use of advanced nuclear magnetic resonance methods to directly measure the mobility of water in living cells contradicts this idea, as it suggests that 85% of cell water acts like that pure water, while the remainder is less mobile and probably bound to macromolecules. 24. Ions, edit. The concentrations of the other ions in cytosol are quite different from those in extracellular fluid. And the cytosol also contains much higher amounts of charged macromolecules such as proteins and nucleic acids than the outside of the cell structure. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Typical ion concentrations in mammalian cytosol and plasma. 7. Ion concentration, millimolar. In cytosol in plasma. Potassium 139 150, 25, 26, 4. Sodium 12 145. Chloride 4 116. Bicarbonate 1229. Amino acids in proteins 138 9. Magnesium 0.81.5. Calcium 0.00021.8. In contrast to extracellular fluid, cytosol has a high concentration of potassium ions and a low concentration of sodium ions. 27. This difference in ion concentrations is critical for osmoregulation. Since if the ion levels were the same inside a cell as outside, water would enter constantly by osmosis, since the levels of macromolecules inside cells are higher than their levels outside. Instead, sodium ions are expelled and potassium ions taken up by the Na/Kat passe, potassium. Ions then flow down their concentration gradient through potassium selection ion channels. This loss of positive charge creates a negative membrane potential. To balance this potential difference, negative chloride ions also exit the cell, through selective chloride channels. The loss of sodium and chloride ions compensates for the osmotic effect of the higher concentration of organic molecules inside the cell. 27. Cells can deal with even larger osmotic changes by accumulating osmoprotectants such as betanes or trehalose in their cytosol. 27. Some of these molecules can allow cells to survive being completely dried out and allow an organism to enter a state of suspended animation called cryptobiosis. 28. In this state the cytosol and osmoprotectants become a glass-like solid that helps stabilize proteins and cell membranes from the damaging effects of desiccation. 29. The low concentration of calcium in the cytosol allows calcium ions to function as a second messenger in calcium signaling. Here, a signal such as a hormone or an action potential opens calcium channel so that calcium floods into the cytosol. 30. This sudden increase in cytosolic calcium activates other signaling molecules, such as carmodulin and protein kinase C. 31. Other ions such as chloride and potassium may also have signaling functions in the cytosol, but these are not well understood. 32. Macromolecules, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Protein molecules that do not bind to cell membranes or the cytoskeleton are dissolved in the cytosol. The amount of protein in cells is extremely high, and approaches 200 mg/ml, occupying about 20–30% of the volume of the cytosol. 1. However, measuring precisely how much protein is dissolved in cytosol in intact cells is difficult, since some proteins appear to be weakly associated with membranes or organelles in whole cells and are released into solution upon cell lysis. 5. Indeed, in experiments where the plasma membrane of cells were carefully disrupted using saponin, without damaging the other cell membranes, only about 1/4 of cell protein was released. These cells were also able to synthesize proteins if given ATP and amino acids, implying that many of the enzymes in cytosol are bound to the cytoskeleton. 33. However, the idea that the majority of the proteins in cells are tightly bound in a network called the microtrabecular lattice is now seen as unlikely. 34. In prokaryotes, the cytosol contains the cell's genome, within a structure known as a nucleid. 35. This is an irregular mass of DNA and associated proteins that control the transcription and replication of the bacterial chromosome and plasmids. In eukaryotes, the genome is held within the cell nucleus, which is separated from the cytosol by nuclear pores that block the free diffusion of any molecule larger than about 10 nanometers in diameter. 36. This high concentration of macromolecules in cytosol causes an effect called macromolecular crowding, which is when the effective concentration of other macromolecules is increased, since they have less volume to move in. This crowding effect can produce large changes in both the rates and the position of chemical equilibrium of reactions in the cytosol. 1. It is particularly important in its ability to alter dissociation constants by favoring the association of macromolecules, such as when multiple proteins come together to form protein complexes, or when DNA-binding proteins bind to their targets in the genome. 37. Organization, edit. Although the components of the cytosol are not separated into regions by cell membranes, these components do not always mix randomly, and several levels of organization can localize specific molecules to defined sites within the cytosol. 38. Concentration gradients, edit. Although small molecules diffuse rapidly in the cytosol, concentration gradients can still be produced within this compartment. A well-studied example of these are the calcium sparks that are produced for a short period in the region around an open calcium channel. 39. These are about 2 micrometers in diameter and last for only a few milliseconds, although several sparks can merge to form larger gradients, called calcium waves. 40. Concentration gradients of other small molecules, such as oxygen and adenosine triphosphate, may be produced in cells around clusters of mitochondria, although these are less well understood. 41. 42. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Carboxysms are protein-enclosed bacterial microcompartments within the cytosol. On the left is an electron microscope image of carboxysms, and on the right A model of their structure. Protein complexes, edit. Proteins can associate to form protein complexes. These often contain a set of proteins with similar functions, such as enzymes that carry out several steps in the same metabolic pathway. 43. This organization can allow substrate channeling, which is when the product of one enzyme is passed directly to the next enzyme in a pathway without being released into solution. 44. Channeling can make a pathway more rapid and efficient than it would be if the enzymes were randomly distributed in the cytosol, and can also prevent the release of unstable reaction intermediates. 45. Although a wide variety of metabolic pathways involve enzymes that are tightly bound to each other, others may involve more loosely associated complexes that are very difficult to study outside the cell. 46, 47. Consequently, the importance of these complexes for metabolism in general remains unclear. Protein compartments, edit. Some protein complexes contain a large central cavity that is isolated from the remainder of the cytosol. 1. Example of such an enclosed compartment is the protosome. 48. Here, a set of subunits form a hollow barrel containing proteases that degrade cytosolic proteins. Since these would be damaging if they mixed freely with the remainder of the cytosol, the barrel is capped by a set of regulatory proteins that recognize proteins with a signal directing them for degradation, a ubiquitin tag, and feed them into the proteolytic cavity. 49. Another large class of protein compartments are bacterial microcompartments, which are made of a protein shell that encapsulates various enzymes. 50. These compartments are typically about 100–200 nanometers across and made of interlocking proteins. 51. A well-understood example is the carboxysm, which contains enzymes involved in carbon fixation such as rubisco. 52. Biomolecular condensates, edit. Non-membrane-bound organelles can form as biomolecular condensates, which arise by clustering, oligomerization, or polymerization of macromolecules to drive colloidal phase separation of the cytoplasm or nucleus. Cytoskeletal sieving, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Although the cytoskeleton is not part of the cytosol, the presence of this network of filaments restricts the diffusion of large particles in the cell. For example, in several studies tracer particles larger than about 25 nanometers, about the size of a ribosome, 53, were excluded from parts of the cytosol around the edges of the cell and next to the nucleus. 54, 55, these excluding compartments may contain a much denser meshwork of actin fibers than the remainder of the cytosol. These microdomains could influence the distribution of large structures such as ribosomes and organelles within the cytosol by excluding them from some areas and concentrating them in others. 56. Function, edit. The cytosol is the site of multiple cell processes. Examples of these processes include signal transduction from the cell membrane to sites within the cell, such as the cell nucleus, 57, or organelles. 58. This compartment is also the site of many of the processes of cytokinesis, after the breakdown of the nuclear membrane in mitosis. 59. Another major function of cytosol is to transport metabolites from their site of production to where they are used. This is relatively simple for water. Soluble molecules, such as amino acids, which can diffuse rapidly through the cytosol. 18, however. Hydrophobic molecules, such as fatty acids or sterols, can be transported through the cytosol by specific binding proteins, which shuttle these molecules between cell membranes. 61. Molecules taken into the cell by endocytosis or on their way to be secreted can also be transported through the cytosol inside vesicles. 62, which are small spheres of lipids that are moved along the cytoskeleton by motor proteins. 63. The cytosol is the site of most metabolism in prokaryotes, 9, and a large proportion of the metabolism of eukaryotes. For instance, in mammals about half of the proteins in the cell are localized to the cytosol. 64. The most complete data are available in yeast, where metabolic reconstructions indicate that the majority of both metabolic processes and metabolites occur in the cytosol. 65. Major metabolic pathways that occur in the cytosol in animals are protein biosynthesis, the pentose phosphate pathway, glycolysis and gluconeogenesis. 66. The localization of pathways can be different in other organisms. For instance, fatty acid synthesis occurs in chloroplasts in plants. 67, 68, and in apicoplasts in apicomplexa. 69. References, edit. 1 ABC Ellis R.J., October, 2001. Macromolecular crowding, obvious but underappreciated trends. Biochem. SCI. 26, 10597604 do. 10.1016/S0968-0004, 01, 01938-7. PMID 11590012. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 2 Kamek, Richard, Atwood, Teresa, Campbell, Peter, Parrish, Howard, Smith, Anthony, Vella, Frank. Sterling, John, 2006. Kamek, Richard, Atwood, Teresa, Campbell, Peter, Parrish, Howard, Smith. Anthony, Vella, Frank, Sterling, John, eds. Cytoplasmic Matrix. Oxford Dictionary of Biochemistry and Molecular Biology. Oxford University Press. Do. 10.100093/acref/9780198529170.001.0001. ISBN 9780198529170. 3 Lyashovitsky, Carlos, 2015. Human Anatomy and Physiology Preparatory Course, PDF. Open. Educational Resources. CUNY Academic Works, 69. Archived from the original on 23 August 2017. Retrieved 22 June 2021. 4 Lardry, H. 1969. On the direction of pyridine nucleotide oxidation reduction reactions in gluconeogenesis and lipogenesis. In Control of Energy Metabolism, edited by B. Chance, R. Estabrook, and J. R. Williamson. New York, Academic, 1965, p. 245, 1. 5 ABCDE Clegg James S. 1984. Properties and metabolism of the aqueous cytoplasm and its boundaries. J. Physiol. 246, 2 pt2r13351 do, 10.1152.1984.246.2.r133. PMID 6364846. S2 SID 303514111. 6 ABC Kamek, Richard, Teresa Atwood, Teresa K. Campbell, Peter Scott, Parrish, Howard. I. Smith, Tony, Vella, Frank, Sterling, John, 2006. Oxford Dictionary of Biochemistry and Molecular Biology. Oxford, Oxfordshire, Oxford University Press. ISBN 0-19-852917-1. OCLC 2255879597. 7 AB Lodish, Harvey F. 1999. Molecular Cell Biology. New York, Scientific American Books. ISBN 0-7167-3136-3. OCLC 1744314882. 8 Hanstein, J. 1880. Das Protoplasma. Heidelberg. p. 24. 9 AB Hoppert M., Meyer F., 1999. Principles of Macromolecular Organization and Cell Function in Bacteria and Archaea. Cell Biochem. Biophys. 31, 324784 do, 10.1007/BF0273824. PMID 107366750. S2 SID 21004307. 10 Baucher CG, Tobin AK, April 2001. Compartmentation of metabolism within mitochondria and plastids. JEXP. Bot. 52, 356 51327 do, 10.1093/Jexpert/52.356.513. PMID 11373301. 11 Goodacre R., Vaidyanathan S., Dunn W.B., Harrigan G.G., Kell D.B., May 2004. Metabolomics by Numbers, Acquiring and Understanding Global Metabolite Data, PDF. Trends Biotech Knoll. 22, 5. 24552. Do. 10.1016/J.typetech.2004.03.007. PMID 15109811. Archived from the original, PDF, on the 17th of December, 2008. 12 Weckworth W., 2003. Metabolomics in Systems Biology. A New Rev Plant Biol. 54, 66989. Do. 10.1146/anav.r-plant.54.03190.1350014. PMID 145030007. S2 SID 11978884. 13 Reed JL, Vote ED, Schilling CH, Paulson Bo, 2003. An expanded genome scale model of Escherichia coli K12, IJR904 GSM/GPR. Genome Biol. 4, 9R54 do, 10.1186/GB200349R54. PMC 193,654. PMID 1295253. 14 carat Forster J, Familiae, Fu P, Palson B, Nielsen J, February 2003. Genome scale reconstruction of the Saccharomyces cerevisiae metabolic network. Genome Res. 13, 224453. Do. 10.1101/GR234503. PMC 420,374. PMID 125666402. 15 carat Lubefalbs K, 2000. Site architecture and physical properties of cytoplasm, volume, viscosity, diffusion, intracellular surface area, PDF. Int. Revelation. Cytol. International Review of Cytology. 192. 189221. Do. 10.1016/S0074-7696, 08, 60527-6. ISBN 978-0-12-364596-8. PMID 1055328. Archived from the original, PDF, on the 19th of July 2011. 16 carat Ruser, Boron W.F., April 1981. Intracellular pH. Physiol. Revelation 61, 2296434. Do. 10.101150/FISRev.1981.61.2.296. PMID 7012859. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 17 carat Bright, G.R., Fisher, G.W., Rogowska, J., Taylor, D.L., 1987. Fluorescence ratio imaging microscopy. Temporal and spatial measurements of cytoplasmic pH. The Journal of Cell Biology. 104, for 1019. 1033 do. 10.1083/JCB.104.4.1019. PMC 21144443. PMID 3558476. 18 carat AB Verkmaners, January 2002. Solute and macromolecule diffusion in cellular aqueous compartments. Trends Biochem. SCI. 27, 12733 do. 10.1016/S0968-0004, 01, 02003-5. PMID 117962221. 19 carat AB Wiggins PM, 1 December 1990. Role of water in some biological processes. Microbial. Revelation. 54, 443249 do. 10.101128/MMBR.54.4.432-449.1990. PMC 372788. PMID 2087221. 20 carat Fulton AB, September 1982. How crowded is the cytoplasm? Cell. 30, 23457. Do. 10.1016/0092-8674, 82, 90231-8. PMID 6754085. S2 SID 63700250. 21 karat Golid KD, 2000. The State of Water in Biological Systems. Int. Revelation. Cytol. International Review of Cytology. 192, 281302. Do. 10.1000/S0074-7696, 08, 60530-6. ISBN 978-0-12-364596-8. PMID 105538283. 22 karat Chaplin M., November, 2006. Do we underestimate the importance of water in cell biology? Nat. Revelation. Mol. Cell Biol. 7, 118616 10.1038/NRM 2021. PMID 169555076. S2 SID 42919563. 23 karat Wiggins PM, June, 1996. High and low density water and resting, active and transformed cells. Cell Biol. Int. 20, 642935 do. 10.1006/KPA.1996.0054. PMID 89632257. S2 SID 42866068. 24 karat Pisani, Hali B., April, 2008. Cell water dynamics on multiple time scales. Proc. National Acad. SCI. USA 105, 17 6266-71. Bib code 2008 NAS 105.6266p. Do. 10.1073/NAS.0709585105. PMC 2359779. PMID 184366650. 25 karat Thia, SO. The 25th of April, 1986. Potassium Physiology. The American Journal of Medicine. 80, 4 AM 3. 7 do. 10.1016/0002-9343, 86, 90334-7. PMID 3706350. 26 karat Lote, Christopher J. 2012. Principles of Renal Physiology, 5th edition. Springer. P12. 27 karat ABC Lang F., October, 2007. Mechanisms and Significance of Cell Volume Regulation. J. M. Cole-Neuter. 26, 5 Supple 613S623S2, 10.1083/0731572.2007.107196667. PMID 179214744. S2 SID 1798009. 28 karat Sussex F., Skopak C., Brady J., Cicero A., August 2001. Reversible dehydration of trehalose and anhydrobiosis, from solution state to an exotic crystal. Carbohyde. Res. 334, 316576. Do. 10.1016/S0008-6215, 01, 00189-6. PMID 11513823. 29 Crow JH, Carpenter JF, Crow LM, 1998. The role of vitrification in anhydrobiosis. A new revelation. Physiol. 60, 73103. Do. 10.101146/anov.physiol.60.1.73. PMID 9558455. 30 Berridge MJ, the 1st of March 1997. Elementary and global aspects of calcium signaling. J. Physiol. 499. PT 2291306 10.1113/physiol.1997.sp021927. PMC 11599305. PMID 9080360. 31 Kikawa U, Kishimoto, Nishitsuka Y, 1989. The protein kinase C family, heterogeneity and its implications. A new revelation. Biochem. 58, 3144. Do. 10.101146/anov.by.58.0701890000335. PMID 2549852. 32 Olaf SN, Harm AP, April 2006. Intracellular monovalent ions as second messengers. J. Memb. Biol. 210, 316172 do. 10.1007/00232-006-0857-9. PMID 16909338. S2 SID 26068558. 33 Hudder, Nathanson L., Deutscher MP, December 2003. Organization of mammalian cytoplasm. Mol. Cell. Biol. 23, 24 9318 26 do. 10.101123.24.9326.2003. PMC 309675. PMID 146455541. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 34 Huser J, 2002. Whatever happened to the microtrabecular concept? Biol Cell. 94, 956196. Do. 10.1016/S0248-4900, 02-00013-8. PMID 1273247. S2 SID 457925524. 35 Thanbikler M, Wang S, Shapiro L, 2005. The bacterial nucleate, a highly organized and dynamic structure. J-cell biochem. 96, 350621 do. 10.1002/JCB.20519. PMID 159888757. S2 SID 253555087. 36 Peters R, 2006. Introduction to Nucleocytoplasmic Transport. Xenopus Protocols. Methods in Molecular Biology. Volume 322. pp. 23558. Do 10.1000/9781597450003 ISBN 978-1-58829 362-6 PMID 167399728 37 Ju HX, Rivas G, Minton AP, 2008. Macromolecular crowding and confinement, biochemical, biophysical, and potential physiological consequences. Anu Rev Biophys. 37, 37597. Do 10.1000/146/37.0328007.125817. PMC 2826134. PMID 1857387. 38 Norris v. Den Blauw Wenti, Cabin Flamino, March 2007. Functional taxonomy of bacterial hyperstructures. Microbiol. Mol. Biol. Revelation 71, 1-2-30-53 Do 10.1000/MMBR.00035-06. PMC 1847379. PMID 1734753. 39 Wang Square, Wei C, Zhao Ji, April 2004. Imaging microdomain CA2+ in muscle cells. Circ. Res. 94. 81011-22 Do 10.1001/res.00001258883.68447.1. PMID 1511789. 40 Jaf LF, November 1993. Classes and mechanisms of calcium waves. Cell calcium. 14, 10736. 45 Do 10.1016/0143-4160, 93, 90099R. PMID 81311190. 41 or, TY. 2000. Intracellular compartmentation of organelles and gradients of low molecular weight. Species. Int Rev Cytol. International Review of Cytology. 192, 22353. Do 10.1016/S0074. 7696, 08, 60528-8. ISBN 978-0-12-364596-8. PMID 105532881. 42 Weiss JN, KGP, 20 July 2001. The cytoplasm, no longer a well-mixed bag. Circ. Res. 89, 2. 10810. Do 10.1161/res.89.2.108. PMID 11463714. 43 Sri Appa, 1987. Complexes of sequential metabolic enzymes. Anu. Revelation. Biochem. 56, 89124. Do 10.1001/.by.56.0701870000513. PMID 2441660. 44 Paham N, 2000. Swinging arms and swinging domains in multifunctional enzymes, catalytic, machines for multi-step reactions. A new revelation. Biochem. 69, 961 1004. Do. 10.1146/anov.biochem.69.1.961. PMID 109666480. 45 Huang X, Holden HM, Rorschel FM, 2001. Channeling of substrates and intermediates in enzyme catalyzed reactions. A new revelation. Biochem. 70, 14980. Do. 10.1146/anov.biochem.70.1.149. PMID 11395405. S2 SID 16722363. 46 Mowbray J, Moses V, June, 1976. The tentative identification in Escherichia coli of a multi-enzyme complex with glycolytic activity. Ur. J. Biochem. 66, 12536 do. 10.11/J.1432. 1033.1976.TB10421.X. PMID 133800. 47 Srivastav DK, Bernhard Sa, November 1986. Metabolite transfer via enzyme complexes. Science. 234, 4781,081-6. Bib code, 1986 SCI, 234.1081s. Do. 10.11/science.3775377. PMID 37753777. 48 Grohl M, Claus NT, December 2003. Molecular shredders, how proteasomes fulfill their role. Car. A pin. Struct. Biol. 13, 6666573 do. 10.1016/J.B.2003.10.005. PMID 14675543. 49 Nandi D, Tahiliani P, Kumara, Chandu D, March 2006. The Ubiquitin Proteasome System, PDF. J. Biosci. 31, 113755 do. 10.1007/BF02705243. PMID 16595883. S2 SID 21603835. Archived, PDF, from the original on 2 July 2006. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 50 Bobic, Tio. 2007. Bacterial Microcompartments, PDF. Microbe. 2. UMSOC Microbiol, 2531. Archived from the original, PDF, on 2 August 2008. 51 Yates 2, Kefeld CA, Heinhorst S, Cannon GC, Shively JM, August 2008. Protein-based organelles in bacteria, carboxysms and related microcompartments. Nat. Revelation. Microbiol. 6, 9681. 691 do. 10.1038/micro 1913. PMID 186791772. S2 SID 226666203. 52 Badger Mr. Price GD, February 2003. Co. 2. Concentrating mechanisms in cyanobacteria, molecular components, their diversity and evolution. JEXP. Bot. 54, 383 60922 do. 10.1093/JXB/ERG076. PMID 12554704. 53 Kate JH, November 2001. Construction of low-resolution X-ray crystallographic electron density maps of the ribosome. Methods. 25, 33038 do. 10.1006/meth.2001.1242. PMID 11860284. 54 Provence DW, McDowell, Marco M., Luba Phelps K., 1 October 1993. Site architecture of size. Excluding compartments in living cells. J-cell SCI. 106, 256577 do. 10.1042/JCS.106.2.565. PMID 79800739. 55 Luba Phelps K., Castle Pay, Taylor D.L., Lanny F., July 1987. Hindered diffusion of inert tracer particles in the cytoplasm of mouse 3T3 cells. Proc. National Acad. SCI. USA 84, 14 4910 3. Bibcode, 1987 NAS, 84.4910 L. Do. 10.1073/NAS.84.14.4910. PMC 305216. PMID 347464. 56 Luba Phelps K., June 1993. Effect of site architecture on the transport and localization of protein. Synthetic machinery. J-cell. Biochem. 52, 2147 do. 10.1002/JCB.240520205. PMID 8366131. S2 SID 12063324. 57 Kholudenko BN, June 2003. Four-dimensional organization of protein kinase signaling cascades. The roles of diffusion, endocytosis and molecular motors. JEXP. Biol. 206, PT 12 2073-82. Do. 10.1242/Jeb.00298. PMID 12756289. S2 SID 18002214. 58 Pasasi P. Schneider, Klein T., Leicester D., December 2007. Intraorganellar communication. CAR. A pin. Plant biol. 10.6606 do. 10.1016/J.B.2007.07.007. PMID 17719262. 59 Winey M., Mama ECL, O'Toole E.T., June 1995. Three-dimensional ultrastructural analysis of the Saccharomyces cerevisiae mitotic spindle. J-cell biol. 129, 6 1601 15. Do 10.1083/JCB.129.6.1601. PMC 2291174. PMID 779037. 60 Wisegera, October 2002. Cytosolic fatty acid binding proteins catalyze two distinct steps in intracellular transport of their ligands. Mol. Cell. Biochem. 239, 1235-43. Do 10.1023/102055040557. PMID 12479566. S2 SID 9608133. 61 Maxfield FR, Mundell M., June 2006. Sterile and lipid trafficking in mammalian cells. Biochem. SOC. Trans. 34, PT 33359 Do. 10.1042/BST03403335. PMID 16709155. 62 Pelham HR, August 1999. The Croonian Lecture 1999. Intracellular membrane traffic, getting proteins sorted. Philos. Trans. ASOC. Lond. B. Biol. SCI. 354, 1388 1471 8. Do. 10.1098/RSTB.1999.0491. PMC 1692657. PMID 10515003. 63 Kamala, Goldstein LS, February 2002. Principles of cargo attachment to cytoplasmic motor proteins. CAR. A pin. Cell biol. 14, 163 do. 10.1016/S0955-0674, 0100295-2. PMID 11792546. 64 Foster LJ, De Hoog CL, Chang Y, April 2006. A mammalian organelle map by protein correlation. Profiling. Cell. 125, 118799 do. 10.1016/J.cell.2006.03.022. PMID 16615899. S2 SID 32197. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 65 Hergard, MJ, Swainston, N, Dobson, P, Dunn, WB, Arger, KY, Arvis, M, Bluthgen, N, Borger, S. Kostenobel, R, et al. October 2008. A consensus yeast metabolic network reconstruction obtained from a community approach to systems biology. Nature Biotechnology. 26, 10.1155-60. Do. 10.1038/NBT 1492. PMC 4018421. PMID 188460889. 66 Stryer, Lubert, Berg, Jeremy Mark, Timochko, John L. 2002. Biochemistry. San Francisco, W.H. Freeman, ISBN 0-7167-4684-0. OCLC 1797059444. 67 Olrog J, Pollard M, Bao X, December 2000. Fatty acid synthesis, from Co. 2. To functional genomics. Biochem. Soc. Trans. 28, 656773 do. 10.1042/BST02805667. PMID 11171129. 68 Olrog JB, Kuhn DN, Stumpf Park, March 1979. Subcellular localization of acyl carrier protein in leaf protoplasts of Spinacia oleracea. Proc. National Acad. SCI. USA 76, 31194-8. Bibcode, 1979 NAS, 76.1194. Do. 10.1073/NAS.76.3.1194. PMC 383216. PMID 286305. 69 Goodman CD, McFadden GI, January 2007. Fatty acid biosynthesis as a drug target in apicomplexan parasites. Cara drug targets. 8, 11530 do. 10.201174/1389450077793155579. PMID 17266528. S2 SID 25652225. Further reading, edit. Wheatley, denies N. Pollock, Gerald H. Cameron, Ivan L. 2006. Water and the Cell. Berlin. Springer. ISBN 1-4020-4926-9. OCLC 71298997. Categories: Cell anatomy, cytoplasm. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.